I'll deal with that in a little bit. An announcement just before we get started. We are going to have a quiz on these last 2 days of stuff. I'll put it up after class just so that you have another opportunity to get some sort of easier points. I'll try and make it an easy quiz. So you'll have a full week to do that. It'll be due on the 7th. So I'll send out an email update and make sure everybody knows about that, but hopefully that'll be good.
You said you were going to put that up after class today.
Yep. And if you guys have something you're traveling for and the timing doesn't work out, shoot me an email and I can fiddle with those deadlines. Key here is flexibility. I just want to give you guys an opportunity to get points that is not during the exams, because I know exams stress everybody out. Okay, so we're going to continue talking about the renal system today. That's not a surprise. So we're going to continue talking about renal function. So when we left off, we started talking about the idea of balance. We'd gone over some terms related to balance, right? Where were we last time? We had talked about the fact that balance is really for regulating blood volume. Okay, sorry, I'm getting a question in the chat saying that the scores for the last weekly quiz aren't showing up. I'll check that after class as well, because those should be visible. Okay, water balance, blood volume, right? That's the main reason we care about water balance is we want to maintain normal blood volume, which allows us to maintain normal blood pressure, allows us to maintain normal concentration of solutes. So this is the reason we care about water balance. So we have these terms that refer to basically whether we have normal amounts of blood volume or high blood volume or low blood volume, hypervolemia and hypovolemia, respectively. And these relate to water balance, because if we have positive water balance, that means we have more water coming in than we have water going out. So we end up with more blood volume. If we have more volume, more water going out, for a negative balance, that leads to a low blood volume. So now we want to focus on exactly how this is happening, like why the water is being excreted, basically, and how we can control the rate at which it is excreted as urine. So whenever we're thinking about water and we're thinking about water movement in the body, we basically want to be thinking about osmosis, right? We know that water diffuses down its concentration gradient, which basically means that it's going to be drawn towards high concentrations of solute, solutes that can't cross the membrane, impermeant solutes, right? So when we're talking about osmosis, we often express things in terms of osmolarity, which is basically a number that tells us like what the pull on water is going to be. So water is going to be moving from areas of low solute concentration to areas of high solute concentration. So as we move through our nephron, we want to be following where we're concentrating solute, because we're going to expect water to follow along. When we concentrate solute somewhere, we're raising the osmolarity, meaning we're raising that like pull on water. So let's take a look. So the osmolarity of normal body fluids is 300 milliosmoles of solute per liter of plasma. So that's our normal value. And what we're looking at right now, grab my pen. All right, so 300. What we're looking at here is a cross section through part of the kidney. So we're looking at the cortex here up at the top. And then as we start to have these gradations, the bottom, this is our medulla, right? So cortex versus medulla. We can see that the cortex is at our normal osmolarity of body fluids of 300 milliosmoles. But the medulla, we can see immediately if we're looking down the side here, right? We can see that it has increasing osmolarity as we go down through it. Now, this is important for water balance and for water reabsorption, because this increasing osmolarity means that we're basically going to be increasing kind of this gradient for osmosis. So we are going to be looking at changes in the osmolarity of the tubular fluid as well. And that'll hopefully kind of explain why this needs to increase, right? Like why we can't just have the medulla be at like 500 and stay there. It is important that we have this increasing value as we go along. So we can see the structures that are going down through the medulla, just so we know. So here's the loop of Henle. So we have a descending limb, descending and going down. We have an ascending limb going up. So it would be seeing a decrease in that osmolarity as it goes up. We have our distal tubule up here by the cortex. And then our collecting duct, this is the part I feel like people kind of forget. This collecting duct is descending back through that medulla as well. So concentrations in the medulla are relevant to the collecting duct as well as to fluid going through the loop of Henle itself. And we'll see that the loop of Henle is actually what regulates, what creates this medullary gradient. Okay. So when we're at 300 milliosmoles, as we are in the cortex here, there's no real osmotic force for water to move between fluid compartments if we stay at 300. So where we're going to see the real forces for movement of water is as we start to descend into the medulla, where we start to see osmolarity above that level, above 300. Okay. So the kidneys are going to compensate for changes in osmolarity of extracellular fluids. So in fluids around their cells by regulating the water reabsorption. So if you start to see variations in the osmolarity of the body fluids, right, the kidneys are going to try to correct that. They're going to do that by changing how much water is in the plasma and therefore changing the concentration of solutes in the plasma, which then goes out to all parts of the body, right? And then we will see water move across the membranes all throughout the body, which is able to correct any changes that we see in osmolarity throughout the body. So water reabsorption is a passive process. So the movement of water itself by osmosis is passive, but we are going to see that in order to set up this gradient in the medulla, right, this gradient in the medulla, which is going to be really important for reabsorption of water because we see part of the loop of Henle going through it, and we see, very importantly, this collecting duct descending through it. We are going to see that we are going to use some energy to set up that gradient. So the energy isn't used for the movement of water, but whenever we see red arrows, right, on our diagram, we know that that's trying to indicate to us that there's some active transport going on. We're going to see that in order to create this gradient that allows water to move via osmosis passively, we are going to do some active transport of solutes in order to control the osmolarity of the medulla. Okay. So if we're talking in just kind of bulk terms, like bird's eye view of where water reabsorption is happening, it's mostly happening in the tubules and the collecting duct, and really the bulk of it is happening in the proximal tubules. So this was the part we've talked about already in a fair bit of detail. This is where 70% of our filtered water is being reabsorbed, right? Everything got filtered at the glomerulus into Bowman's capsule at all the same concentrations it was in the plasma. And then as it moves through that proximal tubule, right, lots of stuff gets filtered out, 100% of the glucose, but also a bunch of water, also a bunch of solutes. So that's not regulated. So that's most of it. Where we do our regulated reabsorption of water, which is going to be most of the rest of the water reabsorption, is in the distal tubules and the collecting ducts. And this is part of why I was emphasizing to you that those collecting ducts are descending through the medulla. So here we see a color difference. They have labeled cortex at the top, medulla at the bottom. All right, so this would be a collecting duct here, descending through the medulla, going down to our renal papilla, into the minor and then major calyces, then the renal pelvis, then going out through the ureter to the bladder. Okay. So as we go through these collecting ducts and the distal tubules, which would be just like kind of the joining point here, this is where our remaining water is going to be reabsorbed. And at this point, as we're going through this kind of final portion of our set of tubes, of our nephron, so the collecting ducts can collect urine from multiple different nephrons. So that's kind of a combined fluid. We're going to see that water reabsorption will continue, but it's going to be regulated by ADH, which is antidiuretic hormone. Okay, and so we are going to talk about that at the end of today. So quick reminder about the water reabsorption in the proximal tubule. So we talked about the solutes being reabsorbed, right? So here we see our solutes being reabsorbed, right? So they're moving into our paratubular fluid and into our plasma and our capillaries. Mainly sodium. Sodium's the main thing that we were reabsorbing, but we also talked about chloride ions, right? They're kind of following along, right? Because those negative charges are attracted to the positive charges in sodium. And then because we're building up these solutes in our paratubular fluid and then in our plasma, water is following along by osmosis, right? Those solutes are exerting a pull, an osmotic pull on our water. So because we have sodium actively transported across the basolateral membrane of our cells in the proximal tubule, this sets up the whole thing, right? This sets up how we move glucose. This sets up how we move water. So in this case, we're emphasizing that it's setting up an osmotic gradient, right? Which is just to say that as we concentrate that sodium, right, sodium adds to the osmolarity. It exerts a pull on the water. So we can look at a combination of what's going on here at the different things that sodium is pulling across, which is now conveniently actually labeled out with the appropriate names of the molecules instead of like the XYZ that we saw before. So basolateral membrane is the part facing the plasma, right? So this is our basolateral membrane. Here's our blood flow in our paratubular capillary. Paratubular fluid refers to this space between, right? So we have a little bit of extracellular fluid between them. Our tubular fluid is over here. So that's what's moving through our nephron, right? So our filtrate's moving that way. Okay. So we can see that in our basolateral membrane, we have a sodium-potassium pump, right? Using ATP to move sodium out, potassium in. Remember, that's an electrogenic pump. What's important for us to realize here in the kidney, right, is that this is concentrating sodium here in this paratubular fluid. So before we talked about it just for sodium's sake, but now we want to realize that this is also raising the osmolarity of the paratubular fluid here, right? Because we're putting a bunch of sodium in it. So that's going to be important for water. But then because we have this concentration of sodium in the paratubular fluid, sodium likes to move down its gradient if it can. Capillary lets it move down its gradient. So we have passive movement of sodium into the plasma. As we were pumping our sodium out into the paratubular fluid, right, that sodium was coming from somewhere. Where it was coming from was the inside of the cell, right? So we were moving the sodium out. So this down arrow is just telling you low. Okay, so we have low sodium inside the cell because we pushed it all out using our pump, which means that sodium can flow down a gradient into the cell towards this area of low sodium. And it can pull glucose with it by secondary active transport. I know we haven't talked about our types of transport in a while, but they're important when we see them here. Okay. So glucose is coming in through secondary active transport here as sodium moves down its gradient. So that is showing us, let me look on this expanded picture, right? So here they've gone back to the Y and X stuff, but here we can see those things we were just talking about, right? So here is our sodium potassium pump. Here is secondary transport of solute Y, which is a little silly because we know solute Y here. They're really telling us what's going on with glucose. Solute X here, right, is just another solute working kind of similarly. This is an actively reabsorbed solute, just something that would work similarly to sodium, right? Where we have active transport across the basolateral membrane, which just kind of sucks things across on the apical side. But the parts we want to think about here as we're thinking about water, which was our real goal, right, is that water is moving by osmosis as these solutes are moving into the plasma, right? So we are concentrating them in the paratubular fluid, which brings water to the paratubular fluid, and then it keeps going into the plasma as well as the solutes continue to go into the plasma. The other thing we have on here is something labeled permeating solute. So what they actually want you to know about is urea. So I don't know why they don't just put urea on the diagram. But urea is a way of eliminating nitrogen in the body, basically, and we'll talk more about it in a couple of slides. But basically how it works is that there are urea transporters in the membrane, in the proximal tubules, and in some other places. And we're going to see that in these places, they passively allow the movement of urea, which is kind of concentrated here in the tubular fluid. So it's able to move into the plasma here, right? And when we see urea again, we're going to be thinking about urea really as also kind of pulling on water and also adding to osmolarity, which I think is why they tried to slightly add it to this diagram here, just so that you know that there is some stuff going on with urea in the proximal tubules. It is one of the things getting reabsorbed here. Okay, I don't have to click through these things in weird orders. Okay, we want to talk about the medullary osmotic gradient, right? Because this is really what's setting up all the details of our regulated water reabsorption. Okay, so what we're looking at, right? Cortex, medulla, and we note that our nephron is moving from the cortex down into the medulla, up through the medulla. It's back in the cortex for a little bit here, and then it's going back down through the medulla as it enters a collecting duct. There could be multiple nephrons going into this collecting duct. Okay. So when we look at the osmolarity here, we can see that the osmolarity of the medulla, as I was saying, has this gradient, right? It varies based on how deep you are. So we can see that right here at the border between the cortex and the medulla, the interstitial fluid, so like the fluid between the cells of the medulla, right, is at 300 milliosmoles. But as we move down, we raise the osmolarity more and more and more. So we're getting to about 1400 milliosmoles here in the deepest parts of the medulla on this diagram. So we have low osmolarity near the cortex, right? So low osmolarity like here, and high osmolarity the deeper we go into the medulla, which conceptually we want to understand as we have like a very light pull on water up at the top, right? So we're only going to have a little bit of a pull, and we're getting more and more and more of a pull as we go deeper. So we're going to be thinking about that as we trace the osmolarity of the fluid as well, because we're going to see that as the tubular fluid moves through the medulla, it doesn't maintain its same osmolarity either. We're going to see that that fluid is getting more and more concentrated as it goes down, which is part of why it's important that we have this really high osmolarity in the medulla as we get to the deep parts. So the fact that we have this gradient is really important to how we are able to continuously reabsorb water. So here we're zooming in on the structure of the loop of Henle going through the medulla, because we want to note some things about the direction of flow and what can move across our membranes in these regions. So here on the left, so we're coming out of the proximal tubule, right? So proximal tubule here, and then we start to dive into the medulla. So as we dive into the medulla here, this is our descending limb. So this is the descending limb of the loop of Henle. Okay. So the descending limb is permeable to water. That's really the main thing that is important to it. It's not doing any type of solute transport, right? What can move across that descending limb, the membrane of those cells, is water. As the fluid continues through the nephron, it goes back up towards the cortex. So this is the ascending limb of the loop of Henle. So it's going back up to where it just was. And you can see that this sort of upper part of that ascending limb is thicker in diameter than either the descending limb or this first part of the ascending limb. So this is the part of the ascending limb that we're really going to concentrate on. This is called the thick ascending limb, right? So it's thick, saying that it's wider in diameter here. And the thick ascending limb is really important because it is a transporter of our solutes. So we can see that we have active transport of potassium, sodium, and chloride ions in the thick ascending limb. So they are being actively pumped into the medulla using the ascending limb of the loop of Henle. The other thing to note here, right, is that if we think about the water, right, the thick ascending limb is not permeable to water, right? So water cannot move through. So water can only cross the membranes in the loop of Henle in the descending limb, and then solutes are actively pumped by the ascending limb, but water cannot cross in this area. And this is important because this is how we set up this gradient. So we're going to walk through how you end up with this gradation, this increasing osmolarity in the medulla. But fundamentally, it's really all being set up by the fact that solutes get actively pumped into the medulla by the ascending limb, and water leaves the fluid in the descending limb as you descend into the medulla. And here we see our friend urea coming out of the collecting duct. So we can also think of this as a solute here, right? So this would also add to the osmolarity here. Mainly we're thinking about the loop of Henle stuff, and that's the part that's a little harder to get your mind around, or at least the part that's more important to get your mind around, I should say. Urea is kind of an add-on topic here. Okay. So we want to think about the establishment of the medullary osmotic gradient. So I'm going to add to our picture just so that we remember, right? Our cortex is at 300 milliosmoles. Our medulla starts out right at this border at about 300, but as you get deeper and deeper, the osmolarity gets greater and greater. So at the depth, we're going to be at like 1400. Okay. So we're setting up this gradient using something called the counter current multiplier. The reason it's called counter current, right, is that we have two things flowing in opposite directions, right? So the opposite part is the counter part, the flow is that current part. Okay, so the counter current multiplier is what's setting up the osmotic gradient. And the opposite flow that we're talking about is the flow down through the descending limb, and then up through the ascending limb. So this is the fluid that's running counter to each other, right? So fluid in the descending limb going down, fluid in the ascending limb going up is really what our counter current is. So it's a counter current multiplier, just to specify that it's kind of multiplying the osmolarity in the medulla. So it's increasing the osmolarity of the medulla, because what we really want to think of the medulla as is like a super salty space that can suck out water. Okay. So it's dependent on the loop of Henle, right? So it's dependent on this opposite flow through our descending and ascending limb. I want to keep our concentration gradient on the side there. Right, so our ascending limb, we said water can't move through here, right? So I'm just gonna write H2O and I'm gonna cross it out. No water movement through the ascending limb, but we have active transport of our ions, right? Sodium, chloride, and potassium. They were a little lazy and just told us the sodium and chloride here, but it's doing all three. But the descending limb is permeable to water. That water is going to move out of the descending limb and it's going to move into the capillaries. So we are going to have capillaries that run alongside the loop of Henle, just like we did along our proximal tubule. Right, we had our paratubular capillaries. We also have capillaries next to the loop of Henle. They're called the Vasarecta. We're going to talk about them separately. And just because I know you've been doing it in lab, they are actually different than the Vasarecta that you see in the intestines when you're in lab. Recta just means straight, right? So they're going to be running kind of straight up and down here alongside the loop of Henle. But we'll look at them separately when it's their turn. So I want you to note, as we're looking at this picture, we're going to look at how this gradient gets set up. I want you to note, just so you know kind of where we're going, that when we start out in the cortex, we start out with fluid, so this filtrate in our nephron at 300 milliosmoles, which is our normal concentration of most of our body fluids. As we descend into the medulla, we can see that osmolarity of the fluid is increasing and increasing and increasing. I want you to think in your mind. I'm going to pause for a second. Think why that would be happening, and then I'll tell you. It's because as we move water out, so we know this is permeable to water, as water moves out into the salty medulla, right, into this medulla that has pretty high osmolarity, this concentrates solutes in the fluid as you get deeper and deeper, right? And this is why it's important that we have really high osmolarity as we get deep down, right? Because if we were at like 500 the whole time, right, that would be pretty salty, pretty high osmolarity relative to our number of 300, but it wouldn't be able to suck water from tubular fluid of 1,000, right? So as we go deeper and deeper, we need more and more of a pull in order to get movement of water. So you'll see that we have increasing osmolarity of that fluid as we go into the loop of Henle and through the descending limb. But then as we track that fluid going up through the ascending limb, we'll see that the osmolarity starts to decrease again, right? So we concentrated up all these solutes, but then as we go through the ascending limb, we're pumping them out, right? So we're pumping out solutes actively using ATP. So this is lowering that concentration of solutes in the fluid again. We can see that actually really lowers it, right? It lowers it to below what we started with through this active pumping. So as we're doing this active pumping of solutes in the ascending limb, we really have two effects. One is that we're making the medulla highly osmolar. We're increasing its osmolarity. We're making it really salty. The other thing that we are doing is we're lowering the osmolarity of that fluid, which we had just increased in the descending limb. So we are going to walk through the setup for how this happens, but I think it's kind of easiest to know where you're going before you look at how you got there. Okay. So this osmolarity in the medulla is the result of the countercurrent multiplier. Our fluid in the proximal tubule starts out at 300. Our osmolarity, as we're going through the descending limb, is increasing because water is moving out of the descending limb. Right? So initially when we start out, our osmolarity in the cortex was the same as the osmolarity in our fluid, right? So the osmolarity of the cortex is the same as the fluid in here. So that interstitial fluid has the same osmolarity as the osmolarity of the fluid. As we move into the medulla, the osmolarity is greater than the osmolarity in the descending limb, which is why we have movement here, right? So like, yeah, say we have an osmolarity of 900 in the medulla. We have an osmolarity of 800 here at this point in the tubule. So we have water move towards the higher number, right? So our osmolarity in the medulla here is higher than the osmolarity in the descending limb here. Okay. So in the ascending limb, the osmolarity of the fluid is going down as it goes up, right? So it's decreasing as we ascend. So we'll go from 1,000, 800, 600. So our osmolarity here is then going to be lower than the osmolarity of the interstitial fluid of the medulla, right? So that same value of 900 was more than the 800 in the descending limb. If we draw that over, right, say we're at like 700 at that same level, I think it would be, right? So our osmolarity of 700 here in the ascending limb is lower than the 900 of the interstitial fluid in the medulla. And it's also lower than that osmolarity at the same level of 800 in the descending limb. So that's what these comparisons are talking about. Okay, so our total results that we're going towards as a result of our countercurrent multiplier of the fact that the descending limb is allowing movement of water passively down its gradient, while the ascending limb, specifically the thick part, is pumping out solutes, making the medulla saltier and saltier. So that we're going to have fluid in the distal tubule at 100 milliosmoles. We're going to have cortical interstitial fluid, so the fluid in the cells of the cortex, or around the cells of the cortex, is going to be at 300 milliosmoles. And the medullary interstitial fluid, so the fluid around the cells of the medulla, is going to be increasing from about 300 at the edge of the cortex, getting greater and greater and greater as we go towards the renal pelvis. So as we go to that sort of curve in the bend of our kidney. So your book lays out the steps that we get here. So this is another one of those kind of like thought experiment kind of setups that we have, like we did when we went through neurons back in A&P1. This is going to start us out imagining everything is equal, nothing's happening, and then we're going to put in piece by piece how we build up a gradient that gets us to our steady state, to our actual observed state in people, which is going to be number 7. So the steady state that you will actually observe in patients, I hope, is number 7. And all these other steps are just kind of adding on piece by piece how we get there. Okay. So we're going to start out by imagining that everything is just at that normal osmolarity of body fluids, right? So here, when we start out in this thought experiment, our cortex is at 300, our whole medulla is at 300, all the fluid in our nephron is 300. Everything's 300. Everything's just at equilibrium, right? So in this state, do we have any reason for water to move? No, right? There's no gradient here in this initial setup for water reabsorption. So we definitely don't want this to happen in our actual kidneys, right? We want to reabsorb water. We want to have an osmotic gradient. Okay. So the first step to setting up that gradient is that we're going to do active transport of ions. So we're going to be actively transporting sodium, chloride, and potassium into the interstitial fluid of the medulla, increasing the osmolarity of these regions. So I'm going to click through to our next picture because that's how we got there. Okay, so we can see on this next picture, right, that because we started this active transport, now if we look at the numbers for our medulla, right, as we did some active transport, we raised the medulla to 400, right? So now we have our medulla at an osmolarity of 400. We still don't see a gradient, but we do see it got higher than the cortex, right? So that's a good start. Okay. We can see that as we pumped those solutes into the medulla, correspondingly, they were coming from somewhere. They were coming from this fluid. So the osmolarity of this fluid went down, right? I have 200 here, used to be at 300 before we were pumping. Okay. So this now means that we do have an osmotic gradient. We do have a force for water to move, right? So if we compare the fluid in the descending limb, 300, to the interstitial fluid of the medulla at 400, water is now going to want to move into the medulla, right? It wants to go water down that highly osmolar area, right? It wants to go water down those solutes that we just pumped. So now we have a force drawn in green for the movement of water, right? So we let that happen. As we let that happen, if we let water keep moving, everything's going to reach equilibrium here between our descending limb and the medulla, right? So we could end up at an isoosmotic state where they're both at 400, right? Now that's not yet where we want to be, right? We got to keep doing stuff, right? But this is our next step, right? We allowed water to move. So now we can see that after we allow that water to move, if we compare the osmolarity of our proximal tubule to the osmolarity of the fluid in the descending limb, we can see that the osmolarity of the fluid in the descending limb is higher, right? So this is kind of showing us how we end up with that increasing osmolarity in the descending limb. Okay. So number 4, now we're going to allow the fluid to move. So before, for these 1 through 3, it was actually kind of just imagining that the fluid was just stationary, just staying there. We didn't have flow. So now we're showing that the fluid is moving. Okay, so more fluid is entering the tubule, right? So plasma is getting filtered out through the glomerular capillaries into Bowman's space through the proximal tubule and now entering the descending limb. So pick a different color real quick. All right, so before we had 400, 1, 2, 3, 4 of them before we got into the ascending limb where we had 200. So now as we see the fluid move, this is kind of like how when we were tracking fresh air moving into the alveoli, we're seeing the fluid from the proximal tubules push into the descending limb. So we still have this section of fluid, but it's moved, right? It got pushed farther along. So we had 4 400s, right? So you can still see them. They're here. Right, so they've been moved farther along. And the fluid that is now in the descending limb is fluid that used to be in the cortex, right? So this was just in the cortex before, but now it's been pushed into the descending limb. So we can see a couple things that we should expect to see in the next couple of pictures, right? So we can see now already, oh, hey, we got a gradient for water transport. Oh, look, they're drawing on continued pumping of these ions. So we can see that we're not done yet, right? We're not at our steady state yet. Okay. So in this state, we have the continued active transport of our ions, sodium, chloride, and potassium, from the ascending limb into the medullary interstitial fluid. So at this point, our medullary interstitial fluid is all at 400. So let's see what happens next after we continue pumping. Oh, I'm going to erase those drawings because this, I wanted to zoom in. We're now looking at kind of the bottom line of the big diagram. Let me clear those. Okay, so this is what happens as we continue, right? So here, I'll highlight for us, right? So here are two of our 400s, right? So we were looking at fluid that got pushed. We were looking at fluid that used to be in the cortex coming down into the descending limb. And we were looking at continued pumping of the solutes. So what has happened here, right, is that we've had water move out of the descending limb by osmosis while we have continued pumping of solutes. So we can see we have kind of two competing things were going on, right? So this used to be, if you look back at the previous picture, this was, these two I think were at 200 and these were at 400. These numbers are now lower because we've had continued pumping of solutes, right? So we went from 200 to 150, we went from 400 to 300, right? Before this was all at 400, but we have water moving through. So we have water coming here and watering down the upper parts of the medulla. And that's still happening as it gets deeper and deeper, but it also gets harder and harder to move water because that fluid is becoming more and more osmolar as it goes deeper. So it doesn't water down as much, but we do still have pumping of solutes. So basically we have more solutes being pumped into the deeper parts of the medulla than we have water moving into them. So we're starting to see this gradient where we have a saltier deep part and a slightly less salty junction here at the cortex. Right. We could then let all of this reach equilibrium, right? Stop moving fluid through it. Everything would reach equilibrium at this state. I say everything, but what I really mean is we'd reach equilibrium on the parts where we can move water, right? So the descending limb would reach equilibrium with the medulla. The ascending limb can't because it's not permeable to water. So it would keep its lower osmolarity because the only way it can move those solutes is by pumping them, and it just can't move water. It's not permeable to water. Okay, so then finally, we just have continued pumping of solutes and continued fluid movement, right? So all of these things would continue to get pushed through the loop. We'd have continued water movement into the medulla. We'd have continued pumping of solutes into the medulla. And if we let this run more and more, we'd eventually lead to an osmotic gradient. It was like this one, but more extreme, right? So we'd have our osmotic gradient of 300 to 1400. And if we look at the actual concentrations, the actual osmolarity of the tubular fluid, it is more extreme than we got up to in step 6 as well, right? So in the descending limb, the tubular fluid in the proximal tubules is at 300. In the turn, where we go from descending limb to ascending limb, it's at about 1400 here. Through the ascending limb, we get lower and lower and lower. We actually get as low as 100 in the distal tubules because of that pumping of solutes out into the medulla. Okay. So our whole big picture looks something like this. So you should understand what is happening in each of these regions, the descending limb, the ascending limb, and why they're important for setting up the gradient, but I wouldn't memorize any of the numbers from these 1 through 6s here. What we want to know, right, for reality, for real patients, is we want to think about the steady state that we're going to be trying to maintain in life, which is number 7. But all these steps are how we got here. So a note about urea. So urea does exist, right? We saw that it was one of the things that's moving across the walls of the proximal tubule. So it's coming from your liver. It's the main way that your body gets rid of nitrogen. And really the reason I care about it, right, is you can't talk about urine without talking about urea, right? So urea is important to the function of the kidneys. It's very water soluble, but it does require transporters. So it can't move across all parts of the nephron. It can't move across just any old cell membrane. It can only move where we have urea transporters. There are 3 of them. Your book probably cares if you know which kind is where, but I am deciding on the spot. I don't care if you know whether it's UA, UTA, UTB, or UTC for our purposes. But there are different types of urea transporters. But what we care about is really where we see them. So we see some in the proximal tubule. We see some in the collecting duct. And then we're also going to see them here in the Vasarecta along this part of the loop of Henle. So this is the Vasarecta, I'll note here. It's the Vasarecta along the ascending limb, but the blood flow through the Vasarecta, through these capillaries that are next to the loop of Henle, is also opposite to the flow of the fluid. So just like the descending limb and ascending limb had flow in opposite directions, creating that countercurrent multiplier, the flow in the capillaries alongside each of them is also opposite to their own flow of direction, which is a little harder to wrap your head around than goes down, then goes up. But it's true. So just be aware of that, because that comes a little bit into play as we talk about the Vasarecta. So I see we're at time, so we will end here for today, and I hope everybody has a good break. But I'm just tracing through, right? So the Vasarecta left the glomerulus through the afferent arteriole. It goes down into the medulla alongside the ascending limb of the loop of Henle, and its own ascending part of the Vasarecta is alongside the descending limb of the loop of Henle. Okay, so we'll pick up here when I see you next week. All right, take care, everybody. Have a great Easter.
Thank you, you too.